Hello. All right, Dr. Ben update for the day. We'll do a couple of updates today, see how much we get time for. The first one is a mathematics paper. Oh my goodness. Yeah. Boy, am I out of my comfort zone. Way out of my comfort zone. Nevertheless, it does appear to apply to some of the results that I've been talking about. And so, yeah. I didn't realize this, but yeah, it makes sense. So here we go. All right, so the paper is called, yeah, this is just such a mathematician paper, yeah. Log parentheses x plus 1 end parentheses star and log parentheses 1 plus x end parentheses star. That is the title, yeah. Are you intrigued? I saw it pop up in the SARS coronavirus section of bioRxiv. That's where I am delving for these little tidbits of treasure, and these are all preprints. So they're things that people hope to publish and have written up, and have probably submitted somewhere, but they are not currently published. They have not yet been peer reviewed. So yeah, so some things change, but this one, I don't know. I don't think this one's going to change much. This is a spicy backhand to the face of several of the other very big, well funded studies that we've talked about here. And so, what this paper is saying is that one problem with all of these single nucleus profiling results that we've been talking about. So, this is where they'll go in and pull out individual cells and then read out the genes inside of a cell. Now, the techniques that we're using are the most efficient that humanity has ever come up with, and yet none of them are perfectly efficient. And inside of a given cell, it's a question of how many copies of a particular messenger RNA are there going to be at a moment in time, which is all you can ever sample. And so the answer is small numbers, pretty small numbers. So the problem mentioned in the paper comes in with the way that we view these things. So for numbers of things in biology, like numbers of bacteria or viruses or genes, you're going to tend to want to use a log scale, which means instead of going 1, 2, 3, you're going to go 1, 10, 100, 1,000, etc., like that. And the problem with a log scale is that sometimes your result is going to be 0. So if you're looking at all the genes that a human can make, a particular cell is only going to be making some of these, and it'll only be making some of them. There'll be a certain amount it has to make before you can even start to detect it reliably with that test. And so there are going to be a lot of zeros there for a lot of different genes. And if you're trying to show 0 on a log scale, which is what we'd like to do, yeah, mathematicians will slap your hand so fast because that's an undefined value. It's it's something like as you raise it to a negative infinite exponent or something, you get kind of close, but you never actually get there. I think that's right, but whatever. Math people, yeah, deal with it. Anyway, the practical point is that so these programs, what they'll do is they will generally add the number 1 to all the results. And so you'll have 1 plus the amount that you detected, or the amount you detected plus 1, yeah. So you can like add the 1 in at the beginning. You can just put 1 in every column and then stack whatever results are on top of it. Or you can have all your results and you could potentially look for the ones that are below 1, and you could make sure that they all end up being 1. Something like that. Yeah, different ways. You can, it's called normalizing. You can do it before or after the test. But for low amounts, so if you found one case, one copy of a particular gene for a SARS receptor, that's where we're getting into the SARS, and you already added 1 to it, then you have doubled the amount of SARS receptor that you think you have detected. Yeah. And so for very small numbers, the adding 1 effect is huge, and this is something that software programs will do automatically, and they do it so that the results don't break and they can actually show you them on a graph that doesn't involve imaginary numbers or whatever the heck the log of 0 would be. Yeah, something that is not real. So yeah, so small values are going to be kind of unstable or unreliable. You would have to use different statistical tests to try and figure out whether your small value of 2 now is actually going to be meaningful, because half of that value comes from the baseline that they put in just so that the software doesn't break. That's the idea. And at the end, there's a wonderful sentence. Let me scroll down and find it. It's the last sentence in this thing. Here we go. These matters are not theoretical. We leave the identification of published preprints and papers that have ignored the issues we raised, and hence reporting misleading results, as an exercise to the reader. So telling you, we're calling out pretty much every single nucleus study. Go have a look at them, see whether they have explicitly corrected for this. I think most of them haven't. I don't recall reading it. And we leave it to you to decide which of these things are now less reliable. So, for example, the one that we talked about with the eye being infected. The corneal cells, if I remember right, were pretty high in ACE2, but the conjunctival cells were pretty low in ACE2. They were actually really tiny. They were kind of at the lower threshold where something like the X plus 1 effect, adding 1, could actually boost that up from something that wouldn't make any difference biologically to something that now looks alarming. Yeah, statistically, mathematically. So there you go. That's a pretty, that's a spicy grain of salt right there to take with all those papers. But yeah, I thought I should bring that to you. It's my duty. Yeah. Thank you very much. This is Ask Dr.